Okay, thank you everybody for joining us today. We appreciate your time and your attention. I'm Iris Flores, Director of Sales Operations at StemBiosis, and we are your hosts today. I'd like to introduce Dr. Travis Block. He is our Senior Technical, our Chief Technical Officer here at StemBiosis. Thank you, Travis.
Good afternoon. So I'm Travis Block. I'm Chief Technology Officer, as Iris said. I've been with StemBiosis for about 5 years, and prior to that was working with cell-derived extracellular matrices in our scientific founders lab at the University of Texas Health Science Center at San Antonio. We've been working with Doctors Heron and Montero de Rocha at the University of Michigan for the last 3 to 4 years on developing new applications of our cell-derived extracellular matrices for getting more biologically relevant phenotypes from iPSC-derived cardiomyocytes. And this work has led to really interesting findings that were published last year in Nature Scientific Reports and have led to some new products for us that I know that some of the people on the call today are using, and I think maybe of interest to more on the call. And we're going to be talking more about those products in the second and third lectures in this seminar series next week and the following week. But we've put together this seminar series as a primer on considerations for the use of cardiomyocytes in vitro cardiotoxicity testing. And with that, I'd like to introduce Drs. Heron and Montero de Rocha. Dr. Heron is the director of the Frankel Cardiovascular Regeneration Core Laboratory at the University of Michigan. In that role, his lab derives new iPSC lines and specializes in cardiac-directed differentiation. Dr. Heron has also developed novel high-throughput screening methods using iPSC-derived cardiomyocytes to detect cardiotoxicity of compounds and environmental toxins. He is the founder of Cardiotox Incorporated, which was based on advances he made at the University of Michigan. Cardiotox has since been fully acquired by StemBiosis. And today, Dr. Heron is the scientific advisor and consultant for StemBiosis, as well as the director of the cardiovascular core. Dr. Montero de Rocha is from Brazilian, but was not a soccer fan, so they kicked him out and sent him up to the US. He got his Doctor of Veterinary Medicine and his PhD in animal reproduction at the University of Sao Paulo. And he worked in human in vitro fertilization and came to the University of Michigan for a postdoc with stem cells. And during that time migrated to cardiology work. Currently, he's a research assistant professor working with Dr. Heron and the StemBiosis team and working on assays and techniques to develop drug discovery applications to bring safe drugs to patients faster and cheaper and and more efficiently. And Dr. Montero de Rocha is also a consultant for StemBiosis. With that, I'll kick it over to the University of Michigan team.
All right, thank you, Travis, for the introduction. It's a pleasure to be here and a pleasure to continue working with the team at StemBiosis. So for the next few slides that I'll be presenting, I'm going to be focusing on the use of human cardiomyocytes for in vitro toxicity screening and preclinical medication and drug development. So I'm going to briefly first go over preclinical cardiotoxicity screening assays, focusing on in vitro assays that are in place now for regulatory science, and give overview of how human cardiomyocytes fit into the paradigm. So it's important to do safety screening of compounds in development for cardiotoxicity, especially focusing on the potential of a compound to cause arrhythmia, called proarrhythmia. So here I'm giving a background on proarrhythmia mechanisms and what's happening at the cellular level when there is cardiotoxicity. So in panel A, you see a normal ECG, an electrocardiogram with the characteristic waves labeled, P wave for atrial depolarization and activation. The QRS complex denotes ventricular activation, and then the interval between S and the T wave is called, or the Q and the T wave is called the QT interval, And this interval is used as an index to determine if there might be an arrhythmia caused by a drug. At the cellular level, what happens during the phases of the EKG, focusing on ventricular activation during the cardiac action potential shown in panel B. So when the ventricles activate, there's a change in membrane potential, goes from very negative potential at rest during phase 4, and through sodium ions entering the cell, there's depolarization of the membrane potential with subsequent repolarization due to activity of potassium currents. And this whole duration of the action potential is referred to as the APD. And the APD correlates to the QT interval. And so the length of the APD determines the QT interval. For medication screening, the screening is done to determine if a medication or a drug in development has any effect on the QT interval. So shown in the top panel, the solid line shows a normal QT interval, and then the dotted line shows the effect of a medication that would prolong the QT interval, where the T wave occurs in a longer period of time. So normal QT is shorter, of course, than a prolonged QT. And at the level of the cells, if we look at the action potential, the electrical activity at the cellular level, any drug that prolongs the action potential duration, again shown by the dotted line, may cause arrhythmias, shown here to the right as EADs. In the EKG, the occurrence of arrhythmias is called EBs, or extra beats, that occur when the heart should be at rest. So safety screening since 2005 has focused largely on determining if a molecule prolongs the QT interval or prolongs the action potential duration. And much of this has been done in vitro using genetically modified heterologous cell systems and an assay called the HERG channel assay. In the cardiac action potential, the repolarization phase or the relaxation phase is caused a lot by the activation of potassium channels and the movement of potassium ions through the HERG channel. Any medication that blocks the HERG channel, as shown in panel C, to block the current of the HERG channel, is predicted to prolong the membrane, the action potential duration at the membrane. And this will cause QT prolongation of the electrocardiogram. So the HERG channel is an ion channel assay performed by many contract research organizations. The HERG contributes to the action potential duration, as shown in panel C, and compounds that block the HERG ion channel can also prolong the APD, and this is assumed to also prolong the QT interval and cause arrhythmias. And because of its position in the regulatory scheme of determining a medication's pro-arrhythmia potential, the HERG assay is labeled as the gatekeeper assay for cardiotoxicity safety screening. However, there is a problem with relying on just the HERG assay, and that problem is that using just QT prolongation or APD prolongation and the HERG channel assay endpoints is that not all HERG channel blockers prolong the action potential duration. So the plot that you see in panel A, this shows relative HERG block on the y-axis versus relative action potential duration prolongation on the x-axis. And you can see for some compounds, as the amount of HERG channel block increases, so does the extent of action potential prolongation. However, there are many compounds, such as the triangle dots here for fluorescence, which is Prozac. You can see that even though it does block the HERG channel, there's very little effect on the action potential duration. So this means that compounds that may fail the HERG assay or that may block the HERG ion channel may not necessarily cause proarrhythmia and may not be unsafe drugs. However, in the current paradigm, compounds that fail the HERG assay or are shown to block the HERG ion channel are often discontinued from further development. And this may be an instance of throwing the baby out with the bathwater, where a compound may actually not be pro-arrhythmic, but the HERG assay mislabels a compound as pro-arrhythmic. And panel B shows that reliance on just one ion channel, the HERG ion channel, is an oversimplification of a complex problem. So the cardiac action potential, as shown in panel B, is the result of the coordinated activity of many ion channels, including sodium channels, potassium channels, and calcium channels. And many medications that may block IKR or the potassium channel may also have effects on the other ion channels, which balance the overall effect to block the HERG ion channel. Another major limitation of the HERG assay is that it's most typically performed using heterologous cells that do not resemble human heart muscle cells. Oftentimes, this may be a CHO cell, which stands for Chinese hamster ovary cell, that do not contract and that only overexpress a single ion channel. So there are alternatives to using the HERG ion channel assay for preclinical toxicity testing, and the optimal preparation is the human cardiomyocyte that will actually be treated by or be exposed to medications that are in development. So there are multiple sources of human cardiomyocytes. One source is primary human cardiomyocytes, which are isolated from human hearts. There are pros to using these cells. A major pro is that they're from a human, so they're the same type of cell that will be treated and exposed to medications. However, there are also many cons to using the primary human cardiomyocytes, and they're listed here. One drawback is that the primary cardiomyocytes are terminally differentiated, so they cannot be expanded. They're available in low numbers, and so this leads to a low degree of rigor and reproducibility using these cells. There will be donor-to-donor variability, enabled to source enough cells. Multiple donors often have to be used in a single drug discovery project. The source of the human hearts may be of question. Most of the human hearts will be diseased or will be from non-failing donors, hearts who aren't acceptable for transplantation into another patient. The isolation of the primary human cardiomyocytes is very difficult and highly variable from heart to heart. The phenotype analysis enabled by the human cardiomyocytes is limited to the single cell level. The cardiomyocytes from the human heart, once they're disassembled, you can't put them back together to form monolayers or to study electrically and mechanically connected cardiomyocytes. In primary culture, human primary cardiomyocytes are unstable. They will de-differentiate over time in culture. And so this makes any kind of genetic modification of the cells difficult and almost unfeasible. Another source of human cardiomyocytes are human stem cell derived cardiomyocytes. These are lab generated cardiomyocytes from human stem cells, most typically pluripotent stem cells. So these cells, there are many pros to using these cells. These cells are from humans. They can be grown to resemble adult primary cardiomyocytes. And because they're created from stem cells, they can be produced in virtually unlimited quantities. And because they can come from a single donor, this increases the reproducibility and rigor of assays for toxicity, because the same cells from the same person can be generated time and time again and used consistently in drug discovery projects. The human stem cell derived cardiomyocytes also can be cryopreserved and used at the discretion of each laboratory. And this really helps to ease workflows of cardiotoxicity assays. Because the human stem cell derived myocytes can be kept in culture long-term and are derived from stem cells, genetic manipulations of the cells are also possible using approaches such as gene editing, like CRISPR. And this is really powerful in order to validate and verify genomic results from GWAS studies, for example, where genetic modifications are identified as being linked to a disease. Here, using these cells, we can start to recreate those genetic abnormalities in a cell line and then determine if there is any effect of those genetic modifications or whether there's no effect of the genetic modification. With human stem cell derived myocytes, long-term culture is possible, and this enables the study of chronic effects of medications. Some medications are given to patients over a chronic period of weeks, months, to even years. So here, using these cells, we can determine the effects of chronic drug administration on human cardiomyocytes in vitro. A really powerful aspect of the human stem cell derived myocytes is that they can be engineered into two-dimensional monolayers where the cells actually couple to one another electrically and mechanically, just as they do in vivo in the working heart. And the cells also, besides using them as two-dimensional monolayers, can also be used to create three-dimensional engineered heart tissues, which can be used also for toxicity testing. The nature of using stem cell derived myocytes is highly amenable to high throughput screening. And excitingly, since the human stem cell derived myocytes can be derived from individual patients, these cells can be used to develop clinical trials in a dish assays so that medications can be screened against multiple populations rather than from a single population or a single patient. And importantly, human stem cell derived cardiomyocytes have been validated in international multi-site studies sponsored and coordinated by the FDA and HESI committee for use in preclinical safety screening. The cons, one drawback of the cells historically and a criticism of the cells is that they are immature and fetal-like cells. So HIPS-CMs, I recommend are preferred for ethical reasons. They're derived from a patient rather than from an embryo. So there are two types of human stem cell derived myocytes that can be generated: embryonic stem cell derived, which you see in the right for the H1 cell line and an H9 cell line, or the cells can be derived from induced pluripotent stem cells, which alleviate ethical concerns surrounding the use of embryos and the destroying of an embryo for research purposes. So these cardiomyocytes, since they were first derived in Tim Camp's lab in 2009, it's been shown that these cells can create nodal SA node cells, which are the pacemaker cells of your heart. Also, we can make atrial cells and ventricular cells. And panel C shows, importantly, that the iPS cells, just like ES cells, respond to positive chronotropic drugs such as isoproterenol. Here where you see the beat rate going from control to speeding up with isoproterenol, which mimics an exercise stress test. But like I said, and as you can see from the images here, long-standing criticism of these cells is that they are fetal-like and not adult-like cells. So this is the new paradigm for the generation of human cardiomyocytes using iPS cells. So somatic cells can be isolated from a donor. These adult cells, which might be skin fibroblasts or blood cells, can be reprogrammed using the classical Yamanaka factors. Dr. Yamanaka discovered the factors required to make cells pluripotent. And then these iPSCs, the iPS cells, are virtually identical to embryonic stem cells, and they can be differentiated into different cell types. For cardiomyocytes, these would be cardiomyocytes, which can be grown in vitro and tested for cardiotoxicity using high throughput screening methods. And the hope here is to be able to deliver safe and effective drugs to patients. So the use of iPS-CMs also is critical for the FDA's movement to reduce the dependence on animal research and to address the 3 R's of animal research to reduce, refine, and replace the dependence of animals for research. And these cells are being used for drug discovery, toxicity screening, and as I mentioned before, FDA safety regulatory guidelines are adopting the use of iPSCs, and the FDA is now accepting data from iPSC-derived cardiomyocytes in investigative new drug applications. So going to the criticism of the cardiomyocytes derived from stem cells, them being fetal or immature. In the work that my lab has done with the team at STEM Biosys, we have addressed this problem by using an optimal extracellular matrix, which is produced at STEM Biosys and is available for laboratory research from STEM Biosys. So what you see in the figure here, these are single cardiomyocytes. On the left, where the cells are circular, these are cells that are cultured on a standard cell culture resource called Matrigel, which is an ECM isolated from a mouse cell line. And you can see human cardiomyocytes cultured on Matrigel take on a circular shape where the sarcomeres, which are the functional unit of contraction in the cells, are disorganized and shaped into a circular shape. On the other hand, same stem cell-derived myocytes cultured on Matrix Plus, which is a product from STEM Biosys, you can see a much different morphology of the cardiomyocytes. The cardiomyocytes take on a rod-shaped morphology. The cells become multinucleated, which is an indication of maturation. And the cells have very well-ordered and very well-structured sarcomeres, indicated by the troponin I staining. Troponin I is a part of the myofilaments that are responsible for contraction of the heart. Also, if we stain for cardiac troponin T, another component of the protein complex responsible for contraction, and also when we stain for alpha-actinin, another part of the cardiac sarcomere, we consistently see that the cells grown on Matrix Plus mature and start to look like primary cardiomyocytes. So these are primary cardiomyocytes. I took from a website from a company that uses primary human myocytes, and you can see that the primary human myocytes are rod-shaped and have very well-ordered and well-structured sarcomeres throughout the cell, similar to the human stem cell-derived myocytes that we grow on Celvo Matrix Plus that you see here. So the long-standing criticism of the human cardiomyocytes being immature and not suited to recapitulate the adult situation has been solved using the optimal ECM to promote the maturation of the cardiomyocytes. So these are images showing that we also can make monolayers of adult cardiomyocytes. So here, this is part of a monolayer where you can see the alpha-actinin staining and the very good organization and parallel alignment of sarcomeres from cell to cell. And in the phase contrast image, you can see over a larger field of view that the cardiomyocytes, when cultured on Matrix Plus, begin to turn rod-shaped and align relative to one another so that they work parallel to each other and also more closely mimic the adult heart and the way that the adult cardiomyocytes are arranged with each other within the working heart. And so that's the conclusion of my slides presenting human cardiomyocytes and their use in toxicity testing. So now I'll turn it over to my colleague, Dr. Montero de Rocha, to discuss subtypes of cardiomyocytes.
And Dr. DeRocha, you're still on mute and outside of presenter mode, just so you know. And while he goes to share his slides, I just want to remind everyone that you can type questions into the chat. And at the end of the session, I'll be happy to read those questions aloud for comment by Drs. Heron and Montero de Rocha.
Okay, thank you, Todd. I just got a message from PowerPoint saying that my presentation is unstable and it just crashed, which is, I'm going to stop sharing this and I'm going to restart my PowerPoint.
Sure. In the meantime, while he pulls that back up, I'm happy to open the floor to any questions. I'll also just comment that in next week's seminar, we'll be giving additional details on the use of Salvomatrix Plus for cardiomyocyte culture and the advantages of that system and how to, what considerations to take into account when looking at extracellular matrices for cell culture and specifically related to cardiomyocytes. So Todd, we do have a couple of questions in the chat from Dr. Obejero Paz. Do cells in Salvomatrix Plus show mature T tubules?
You're muted, Todd.
Sorry, Carlos, that's a good question. We haven't done the careful T-tubule assessment yet. That's next on our agenda of things to do. We plan to use dye-8-NEPs to do in vivo live cell T-tubule staining, and we'll probably also try WGA staining.
And then from Dr. Smuckler, Smookler, will the cardiomyocyte maintain the mature phenotype if we move them to a new substrate?
That's also a good question that we haven't tested yet. So we have focused on phenotyping the cells as they are. We haven't gone on to re-aggregate or to split the cells onto a new matrix yet. So that we don't know either.
That is work that's planned both at the University of Michigan and at our labs in San Antonio at STEM Biosys. So we're looking at cryopreserving mature cells and seeing if we're able to maintain a mature phenotype after reseeding. And so hopefully by the next time you see a seminar like this, we'll have an answer for that question. With that, I'll turn it over to Dr. Montero de Rocha.
All right. Well, thank you for your patience, and I think that now we're able to go ahead and see the slides that I've prepared. So as Todd mentioned in his presentation, when you generate cardiomyocytes from iPSC cells, in general, using the most common protocols, you can get cells that have an atrial identity, a ventricular identity, and also nodal cells on the same batch. And okay, so all right. So, but more recently, there were some other protocols that started to be published that they try to produce cells that are ventricular specific or atrial specific, depending on what are the cues that you give to the cells during the process of differentiation. So as I've mentioned before, when you get cells, cardiomyocytes from a regular protocol, you're going to get cells that have ventricular cells and atrial cells. So this is some work that Todd's lab performed in 2013, and it was like an early attempt to start sorting out atrial and ventricular cells from this general pool of cells. So at that time, they used an adenovirus that was expressing GFP driven by either the myosin light chain 2A, which is the atrial isoform, or 2V, which is the ventricular isoform. So they used those cells and applied to I-cell cardiomyocytes. And what they found is that there was an expression of about 38.5% driven by the atrial isoform, expression of GFP driven by the atrial isoform. And from the same batch of cells, when they were using the other adenovirus that would drive the GFP expression with the MLC2V, which is the ventricular isoform of the myosin light chain, they would get about 43.5% of cells expressing GFP, and they've used that to use flow cytometry sorting to generate populations that were pure atrial or pure ventricular cells. Although it was like an ingenious way to do that, we all know that passing the cells through the flow cytometer and selecting them in this way can be, it's not very, I would say not very efficient, and you would end up losing a lot of cells, and you also have to do the adenovirus transformation of the cells, and it didn't prove to be like very practical, but it was an early attempt to start addressing the chamber specificity of the cells. There's also another thing that, at least with the cells that we generate in-house, we also tend to see a lot of cells that express both isoforms of the myosin light chain. They express the atrial and the ventricular isoforms of myosin light chain. And as Todd mentioned before, the action potential that we're going to be using as a surrogate for the QT interval, it depends on several different ion channels, and these ion channels will be expressed in different ways or in different densities depending on the cell type. If it's an atrial cell, if it's a ventricular cell, there are also, like Carlos asked about, like the T tubules, and the depth of the T tubules and location of channels are going to be different between atrial and ventricular cells, and that can greatly impact the response of the cells to different drugs. So because of that, we decided to start here at the University of Michigan making chamber-specific cells. We did not develop our own protocol, but we tried several protocols, and the one that worked the best for us was published in JCI Insight in 2018. And the protocols are very similar, but there is a key component that is retinoic acid that when you add it on day 3, between day 3 and day 6, it actually drives the cells to differentiate into an atrial fate. So for those that are not very familiar to the differentiation protocol, at day 0, you have a monolayer or a single layer of stem cells, and then you apply a GSK3 inhibitor that's a CHEER, and then this will drive the cells into mesoderm. And when you apply IWP4, which is a Wnt signaling inhibitor, the cells are going to turn into cardiac mesoderm. And then from here, from day 4 on, the cells are going to be specified as cardiomyocytes. So it's very important to start applying the retinoic acid when you have a specification of the cardiac mesoderm, because this is what is going to give the identity of the cells. And at the end of day 8, or when you do your analysis, you would expect to have beta myosin heavy chain on the ventricular differentiation protocol, because this is ventricular marker, and very little or known alpha myosin heavy chain that is present on atrial cells. And of course, that the results expected are the opposite here on the atrial protocol. So here we have some cells that we've generated here, and I want you to see that these are the ventricular iPS-CMs, and you can see that they're beating fairly slow. They also are fairly big cells in comparison here to the atrial cells, in which you have a much faster activation rate. And you can also note that the cells are much smaller in size in comparison to the ventricular cells. And these can have like really important impacts. For example, when you're challenging the cells with isoproterenol and some other drugs that alter the frequency of spontaneous depolarization. So these are some quantifications from those movies that you've seen. These are action potential fluorescence measurements. So here at University of Michigan, we're specialized in producing optical mapping systems. So to measure either calcium or voltage change using visual voltage sensitive dyes or calcium sensitive dyes that they fluoresce every time that there is a change in action potential or intracellular calcium release. Here you can see that the ventricular cells, they have a much slower activation rate in comparison to the atrial cells. And this has a great impact on the action potential duration. So we quantitated the action potential duration at 80% of repolarization, which would be around here, right? You're measuring how long it takes to get back to almost the resting state. And you can see that in the atrial cells, the action potential at 80% is much shorter than on the ventricular cells. And this is mainly because of the change in frequency. We also started testing some of the biochemical markers. So here you can see that this is some of the data that we generated in-house. And the atrial cardiomyocytes, we cannot detect beta myosin heavy chain, but it's very abundant on ventricular cardiomyocytes, and this is the positive control sample from a gift of life sample. It's basically a ventricular sample from our heart, human adult heart, and this is the quantitation of this difference. So I also brought to you some of those like beautiful stainings from the Siganek paper. So here he has like phalloidine and comparing the control, which would be the ventricular cells and the cells that were treated with retinoic acid for atrial specification. So this is the MLC2V, the myosin light chain 2V, and you can see that is abundant on the cells that express this marker are abundant on the control, that because this is a protocol that generates ventricular cells, but very few cells were positive when you applied retinoic acid. And in a complementary way, the MLC2A expression is very limited on the cells that were treated with the protocol that generates ventricular cells. But you can see that here in the atrial, the cells treated with retinoic acid, you have many more cells that are that express the atrial isoform, and this is just the overlay of the two of them, showing that the majority of MLC2V cells are on the control or ventricular protocol, and the MLC2A expression is much higher on the atrial cells. They also showed, which is like very interesting, because nowadays people are trying to develop new protocols and new tests that include long-term treatment of cells. And they show that these pattern of markers expression is stable on cells after 60 days in culture or 90 days in culture. So you can see that MLC2V expression is present only in the control and not in the atrial cells. They also did a lot of RNA sequencing, and they showed that the ventricular cardiomyocytes, independently of the cell line, they tend to group on a different cluster than the atrial cardiomyocytes. And this is the dendrogram representing these clusters. And again, independently of the cell line that they've used, the cells tend to cluster. And this panel here, this heat map, shows that there are like very significant difference in the relative expression of sodium channels, potassium channels, and different ion channels, which might be very important for drug response. And might change the, just a second. And might change the way that the cells respond to drugs. And this is something that is very important to take into consideration when you're developing a new drug or performing new tests. And it's also important not only for drug screening, but also for when you're studying different disease, because some diseases have more of like an atrial phenotype than a ventricular phenotype, or the other way around. And it's important that you're using the appropriate model to answer your scientific questions. I think that this is the last slide, and I think that we can go back to questions. You're muted, Travis.
While we wait for any questions from the attendees, I actually would like to kick it off. So when you're specifying atrial and ventricular cardiomyocytes, I know that we're seeing spontaneous beating. Are nodal phenotype cells necessary to see spontaneous beating in vitro?
So this is a good question, Travis. So one thing that we always see is that when you form a new monolayer, the pacemaker it is always in the same place. So you can overcome the pacemaker site using an electrode and doing an artificial pacing. And at the moment that you remove that stimulation with a point electrode, and you can only, of course, see that with the point electrode, because if you do a field stimulation, you're stimulating everything at once. But with the point stimulator, let's say that you have your pacemaker at 9 p.m. and you stimulate the cells at 12 hours, right? If it is a clock. And you're gonna see that you can change where the pacemaker is. You turn off the electrode, and the pacemaker will return to the same place. So there is something special about that region of interest, and perhaps it's because you have more nodal cells. If you get these cells and you plate them as single cells, you can see that some of those cells are also going to be having spontaneous beating, but it does not mean that they're nodal cells.
Interesting. So I think I tend to think about nodal cells as regulating the frequency of the beating. So how does it work with atrial and ventricular cardiomyocytes in vitro having different beat rates? Is there some kind of feedback mechanism to nodal cells to help regulate that?
So the atrial cells that Andre showed, they spontaneously depolarize at a faster rate, which suggests that they express more nodal-like ion channels, such as the funny current, which is responsible for the unstable resting membrane potential. On the other hand, the ventricular cells, which beat at a much slower rate, it's likely they have fewer expression of these ion channels. So they don't express as many of the spontaneously depolarizing ion channels. And that's why they have a much slower beat rate. And this is similar to what you would expect to see in cells isolated from the heart, where a healthy ventricular cell would have almost little to no spontaneous beating, although it can still spontaneously depolarize just at a very, very slow rate. And the atrial and nodal cells would be expected to spontaneously depolarize at a faster rate than the ventricular cells.
Sure. And I hate to put you guys on the spot because I had weeks to give you guys questions if I was going to ask them, but I'm going to put you on the spot anyway. Can you tell me about are there any classes of drugs or classes of ion channel inhibitors that we would expect to see an effect in atrial cardiomyocytes that we're missing today because we typically do these assays in ventricular phenotype cells?
So I guess that would be if there were any atrial specific drugs. There aren't very many atrial specific drugs, but I think what's critical is in order to develop any new atrial fibrillation medications, it would be ideal to test those new medications using atrial cardiomyocytes. And so I think for the future of drug development, the use of chamber-specific myocytes, of chamber-specific human myocytes with high throughput screening will help lead to the development of chamber-specific medications with higher specificity and also greater efficacy.
Excellent. Thank you so much. As always, I love having the opportunity to work with both of you, both in these seminars and more broadly on our product development efforts. Just a reminder that if anyone has any questions, you can either speak up or type them in the chat window and I will ask them. And with that, we'll stay on the line for a few more minutes to see if there's any questions, and then we'll hop off, and hopefully we'll see you next week for a more in-depth discussion on considerations for substrates and studying cardiomyocytes and our own use of Selvomatrix Plus to mature iPSC-derived cardiomyocytes.
Thank you, Travis, and thank you, team, for an excellent presentation. Just a reminder, when you registered for the original seminar, you actually registered for all three, so you should have reminders in your calendar. And so the next session is actually October 21st. It's 2 weeks from now, 2 p.m. Eastern Standard Time. And this is going to be focused on the use of cell-derived matrices like our own Selvomatrix Plus for iPS cardiomyocyte culture. We're going to go into a little detail there, and we're also going to talk about the cell types that we've used and some of the data that the University of Michigan has developed using our substrate. If you have any questions about the particular products, you can always visit our website at www.stembiosis.com. You can leave messages for us there. You can learn information about all the products there. You can also, we have a chat option. So if you have additional questions about this, we'd love to hear from you. We're going to post the recording of this here on our website as well, and we'll continue to do that for the rest of the series. We do appreciate your time and your questions today. It was very, this is a great presentation, Todd and Andre, Travis. Thank you. Do we have any more questions from our participants? Okay, I think that if Andre and Todd, if you don't have any additional information, we can end the seminar here. Everyone, thank you again for joining us, and we look forward to seeing you again in 2 weeks.
Thank you.
All right, thank you.
Thank y'all.